Hello, my name is Paula Bourke. I'm a professor within the School of Biosystems and Food Engineering. I'd like to thank you for the opportunity to showcase some of my research during the Institute for Food and Health Research Day this year. So one of my main research interests is in the area of applying cold plasma technology and cold plasma biosciences. So today I'm going to present some of the work we're doing surrounding cold plasma innovations for safe and sustainable food. So we're all familiar with, I suppose, some of the rationale for seeking sustainable food processing technologies. The Food and Agriculture Organization has produced many reports, there's more on the way, demonstrating and providing areas where we need to do better. There's a food loss is a major focus, I suppose, of a lot of the research that we do, whilst we're looking at ways of improving the shelf life or improving the quality of food. This is actually trying to bring food back into use and retain it within the food use cycle, as opposed to losing that resource which started off as perfectly useful food. So one of the things that I do a lot of research on is applying cold plasma, which is a novel non-thermal technology, to resolving some of these problems with regards to food sustainability and food safety. So just to explain a bit about what cold plasma is. It is an ionized gas consisting of free electrons, ions, reactive atoms, neutral molecules, and photons. So if you think of it as you're essentially adding energy to get to different states of matter. So a relatively simple explanation is if we start off here with water in its solid form as an ice cube. When we add a bit more energy, it melts, becomes a liquid. When we add more energy, it becomes a gas in the form of steam. And when we add more energy to that gas, when we supply more energy to a gas, it becomes a plasma. And we end up splitting everything, and we get a huge array of charged particles, free electrons and ions. And those are demonstrated there on that slide for just when we started off with water. So you can see what a diversity of species that are generated, of reactive oxygen, reactive nitrogen species that can be generated from simple gases such as air or simple constituents such as water. So that plasma state can be reached basically by supplying enough energy to that gas or to a mixture of gases. And one of the advantages of plasmas, they can be operated at both low and atmospheric pressures, so they're relatively flexibly incorporated into different processes. It is a flexible technology in terms of its potential applications to the food and agricultural arenas. It can be, I suppose, termed a platform technology, where we could use it from an agricultural perspective. We could harness the reactive oxygen and nitrogen species, or indeed the different actions of applying the technology, such as the electric field or the UV. And a lot of those mechanisms can give very advantageous characteristics in terms of agriculture. We could make a plasma generated nitrate fertilizer. You can increase the germination rate of seeds. You can increase plant yields potentially, depending on the type of plant. You can use it from a kind of a biodecontamination perspective, where you might control the occurrence of pathogens or the prevalence of spoilage of a particular foods. And I suppose a lot of the interest has been in foods that spoil relatively quickly, such as fresh produce, meats, fish, and dairy products. It can also be used and tuned, I suppose, to a certain extent, to degrade things like chemical constituents or residues that may be present on foods. But with the shift away to more sustainable use of pesticides and sustainable approaches in agriculture and food biosystems, the degradation of residues and things that might already be on food is probably, whilst it's still valuable, we'll be more looking at ways that we can utilize the technology to avoid applying chemicals or residues in the first instance. So what I'm going to focus on today is a project that has been going on for a number of years. It's called Plasma Grain. And this project was a multifaceted exploration of applying cold plasma to cereal grains and the issues that prevail for cereal grains, sustainability and safety. And also we tried to develop some cold plasma technological innovations within this project. So I'll try and go through some of the key areas of research that we did within this project. So some of the key research areas within this project, it was, as I said, a multifaceted project. So we had a lot of different focus areas. One of the main ones towards the beginning of the project was on microbiological control of many of the issues that present on grains. The issues can come from the field, they can come from in storage, but how can the technology be applied to best decontaminate grains? And is it safe indeed to do that? We also focused on some pesticides and mycotoxin degradations. We looked at insects control, and we've also looked then at actually developing technology. So we started with the platform technology, and we've built on this to develop a range of technologies, which I'll go through towards the end. But also one of the more interesting areas is that we can actually, we're seeking to bring grades of product that may need a little bit of help in retaining their quality or providing functionality to that grade of product to bring them back into the human food chain. We can also look at the functionality modification of the grains, and there's a lot of investigation, a lot of interest indeed in developing this type of an approach as a pre-sowing seed treatment, and actually seeing is this a potential way that we can deal with a lot of the plant pests that we're kind of running out of solutions for. So the two areas that I'm going to focus on are, within this talk are in that quality retention area and the tailored technology development. So the types of contamination that prevail on cereal grains, we've got them coming from the field and we've got them coming during storage. So there's a huge diversity of risks and issues that we need to deal with. And each of these need to be dealt with really individually. The contamination can come from the field. It can reside in the leaves of a plant. It can be bacterial, it can be fungal, it can come up through the roots, reside in the stem or the heads. So pre-harvest, we were looking at things like, can we improve the seeds' germination properties? Can we enhance the seeds' performance? And does that then translate into plant performance? But then when you've actually harvested your crop, so post-harvest then, within the storage and processing stages, the questions were really, we're still dealing with a lot of microbiological or biological risks. You've still got insects, rodents, bacteria and fungi leading to up to 40-50% spoilage of a particular crop during storage. So what solutions can we provide that will provide us with post-harvest safety and quality retention? And can we improve that harvest usability if some of the products have reduced quality characteristics? So the system that we used for looking at a lot of the work is a contained air-based plasma reactor. And we've used this as a platform technology throughout much of the studies for the microbiological, the biological, the quality. So we have a level playing field for all of our interrogations and our fundamental studies. So we would have placed some seeds within the plasma field, within a container, and exposed to the plasma either directly or indirectly. And basically direct application means you've got the electric field, the UV, as well as all of the plasma reactive species. Whereas with indirect ECP or cold plasma treatment, we're not necessarily exposing the samples to the UV, we're not necessarily exposing them to the electric field, but predominantly they're being exposed to the reactive nitrogen and the reactive oxygen species that prevail within a plasma that is generated using air. So one of the kind of key studies we looked at then was we were looking at wheat and barley predominantly within this project from diverse locations, and we wanted to see what plasma processes can we develop that would actually enhance the germination of these seeds. So how can the cold plasma treatment improve the germination parameters of wheat seeds? So we did some germination studies on the seeds, and we took some measurements. We also looked at what happened to the seed when we treated it. What was the chemistry? What were the responses of the seed to that treatment? So what we found is that by comparison with our control seeds, which weren't treated to anything at all, and we compared the mode of plasma where we used indirect treatment compared to direct plasma treatment. And we did it over relatively short treatment times because these are live systems. So we're talking about impacting enzymes or activating enzymes or activating biochemical and biological systems. So if we treat too long, we might actually interfere with those systems. We want to retain and enhance these quality characteristics. So very short treatment times were applied. And we found that very short treatment times in the range of 30 to 60 seconds can significantly improve the germination properties of the wheat seeds. And then following on from that, when we grew out those wheat seeds, the growth parameters of those seedlings were also enhanced. We got an increase in the shoot length. We got an increase in the roots length, and that can be seen there across. Where on the left-hand side we have the control seedling, just visually represented there by in the images. And as we increased treatment time using direct or indirect mode of treatment, we did actually enhance the root and shoot length, with the optimum results reached afterwards with relatively short treatment times. So we found that indirect treatments, so those treatments utilizing the reactive oxygen and nitrogen species generated within the plasma process, indirect treatments, very short treatment times between 30 and 60 seconds, enhanced germination by around 13 to 14%, which was a very useful impact. So generally speaking then, shorter, much quicker plasma processing treatments are more suitable for germination enhancement and looking at those kinds of quality characteristic retention. Whereas if you wanted to do microbiological decontamination of your grain, if you're not going to use it for seed, you might have to apply a much longer treatment time or a more intense process that would actually potentially have a detrimental effect on the biological activity that would lead to germination, but would actually certainly biodecontaminate the surface of a grain. So the process choice depends on what the final function of the product is going to be. So basically, we were trying to understand what are the mechanisms of germination enhancement by cold plasma, particularly for our cold plasma treated wheat seeds. So we did a series of investigations looking at the surface of the seed, because there would be a potential that we could actually have cracked the seed and that could have led to water uptake. But we found that there wasn't actually any major visible changes using scanning electron microscopy, but we did note huge changes in the surface chemistries. We noted a decrease in hydrophobicity, and we actually got a huge increase in water uptake. So that actually, combined with these chemical pH and chemistry changes, actually led to the enhanced germination properties. So these are fundamental understandings then of the system that could potentially be applied to other seeds and other plant systems. So moving on from that aspect, we also then wanted to look at other quality characteristics and understand what is the influence of cold plasma on the functionality of wheat flowers. So we're starting off with wheat seeds and growing plants, but then we also took those wheat seeds or grains, and the other use then is I suppose, as flours ground down, incorporated into such a wide range of foods. So we would really like to understand what's going on there. So we had our flour samples, again, went into a container, was treated within the direct plasma treatment and also indirectly. So one of the issues with flours is they don't always meet the industry requirements in terms of their quality characteristics. The very high-speed processing lines means that there's actually quite a low tolerance for any variations in flow or behavioural variations in the flour doughs, which depends on their in chemistry from the grains on which they're ground. And there are a lot of agents, approved agents, additives, that can be added in to improve wheat functionality in commercial and retail products. So they might be oxidizing agents or enzymes. Now, there's limitations associated with some of those. Obviously, if you're adding in any chemical to a food system, you are adding in the potential to incorporate any toxicological effect along that food chain, but also adding in an enzyme to improve performance is an expensive way to improve the quality of your product. And then you also have to label any product that you've added a process additive or added an agent to. So these are some of the considerations we were taking into account when we were designing our studies. So what we did was we took our flour, we exposed it to the plasma treatment. And we really wanted to understand what was the influence on the physical chemical properties of the wheat flour, because it is those physical chemical properties that actually are responsible for a lot of the functionality of flours and the proteins and the different constituents of the flour. So we looked at the effect of applying the plasma at 2 different stages. We applied it to the grain. We also applied it to a flour. And we were trying to develop then from that, what is the next step, a little bit of product development. So we chose to try and make something from our plasma treated flour and to investigate what happened, the actual product development stages for that. So we chose bread. And our key findings are, first of all, looking at the plasma treated flour, where we took the grains, we then ground it down, or we had treated it and then we ground it down and then looked at many of its physical chemical characteristics. So the water hydration capacity of all of the plasma treated flour samples actually increased with increasing treatment time, which is also, with regard to the system we used, really increasing process intensity. An increase in the water holding capacity of a flour can be basically due to the hydrolytic depolymerization of the starch within that flour. We also then looked at the differential scanning calorimetry. And again, the plasma treatment affected the gelatinization temperature, which can be a useful characteristic. And an increase in the peak temperature can actually be attributed to the cross-linking that happens on the surface and the internal matrices of those starch granules. We also looked at the whiteness index, and we actually got an increase, a small increase in the whiteness index, but only after prolonged treatment, after 30 minutes of treatment, of consistent treatment, which is quite a long time. But that can be then attributed to degradation of conjugated double bonds of the formed from the carotenoid pigments, potentially by ozone species within the plasma treatment process, or the retention of ozonoid particles as well. In terms of the viscosity, we got a significant increase in the peak viscosities and the final viscosities as a function of treatment time. So that actually may be leading to a loosening of the starch granules. And this then was confirmed with our X-ray diffraction, where we saw a change in the overall intensity of the peak for all of the plasma treated samples. So again, indicating that we're depolymerizing the starch granules. So the next step then was to actually take this the next step further and actually look at product development of a white pan bread from our plasma functionalized wheat flour. So we weren't just treating them at this stage. We'd established what plasma process parameters we wanted to apply. So we chose those and we treated a specific batch of our starting material, our wheat flour. And we went through a product development process by comparison with control and normal retail breads, and actually made a bread. And this image that you can see on your left is actually a micro tomograph of the bread, of the loaf of bread, of a slice of that loaf of bread. So the various steps that we did were, we call this the plasma bake experiment. So we treated our flour, then we have plasma functionalized flour, and then we went through our bread making procedure. So we have plasma functionalized flour bread. So we were looking at things like the color, the crust formation, the how did it rise as well as a normal untreated flour, and what did the inside of the bread look like. So we had a few different ways of approaching this. So we looked at the expansion ratio, the water activity, and again, we did some basic sensory analysis on this. So what we found is that the expansion ratio of the dough increased with plasma treated flour. And industrially, many leavening agents are actually used to get this effect. So it is potentially a way of replacing that type of additive to your product. The water activity of the bread increased with plasma treatment, and this improved the water intake capacity of the flour. And that actually gave us a very moist bread, which was quite a desirable thing. And then in terms of the sensory analysis, what we found was that overall, it was a very general sensory analysis was done. The acceptance of this bread made with the plasma functionalized flour scored very comparably to the in-house control, which hadn't been plasma treated, or was bread made from normal flour that hadn't been plasma treated, but it actually exceeded the retail bread sample. So it's a very encouraging result And we're certainly looking forward to doing more of these types of experiments. So this is what the inside of bread looks like when you take images of it. And the white represents the actual bread formation, and the dark then represents the air voids. So as you can see, there are changes. There are actual discernible changes within the amount of voids and the distribution of those voids within the dough in relation to the plasma treatment, which is can give us some very desirable effects. So again, here, this is the X-ray microtomography images of those bread. So coming down the page or coming down the slide, you can see this is it's certainly when we're applying or increasing our plasma process and moving across in the middle column there. We're looking at the size distribution of the voids, and that changes. You can really see that changing. Those patterns change with increasing plasma process intensity. And then towards the other side, to the right-hand side there of the slide, we've got a 3D reconstruction of the air voids and different angles of those. So this is under analysis to actually see can we model the plasma process effects on the air voids within bread. Another important area with regard to wheat and grains is very many people are actually have a wheat protein allergy in relation to gluten, or they have an intolerance for it. Cold plasma is a technique that can actually, part of its effects is that it can modify protein structures and amino acids composition. And this is it. We've gone through this in a recently published paper in Food Chemistry for dairy, for milk-based protein allergens. But this study we've started now looking also at the wheat proteins and glutens. Celiac disease is actually quite prevalent. Approximately 1 in 100 people in Ireland have celiac disease associated with gluten intolerance, and there's also then gluten allergies. And there's various unpleasant symptoms, but not necessarily saying that the technology may be applied to deal with changing the allergenicity of a food product, but if we can actually modify the proteins and reduce the allergenic epitopes and understand how that can be done, there's potential that we can apply this approach for cross-contamination control and mitigate contamination in food processing environments with allergenic residues. So it can be a very useful thing to understand. So the wheat protein allergenicity is caused by this. Wheat gluten, is comprised of gliadins and glutenins. And we have different structures, different molecular weight, different amino acid residues. So we'll be investigating to see what happens these parts and these physical structural characteristics of the wheat proteins. So the plasma gas phase has a wide range of reactive oxygen species that could potentially interact with those proteins and break those bonds. We might be able to oxidize them, and there are a wide range of mechanisms by which cold plasma can actually interfere with protein structures and protein bonds. So we'll be investigating these for further understanding on how we can actually control the wheat proteins. We have established how we might be able to control the dairy proteins allergenicity, and now we're moving on to wheat proteins allergenicity. And one of the issues with wheat proteins is they're actually relatively heat stable. So there is a need for some process interventions that might actually be able to deal with these cross-contaminating allergens. At the moment, are a sequence of reactors, of plasma reactors that can actually utilize air instead of a noble gas like helium, or we can do combinations of gases. And we're up to a stage where we can treat kilograms of grains and seeds using a plasma bioreactor, which is a different design now to the auger system, but it is most certainly a barrel and a system that can be used in conveyance or batch or continuous treatment. So we're at the moment we're investigating this. And one of the other key questions we're addressing is using cold plasma to treat grains a biologically safe thing to do? So we've had many experiments, I suppose, trying to understand this aspect, and what we came up with was a completely different system which actually looked at a controlled mode of plasma discharge to water, because that would actually allow us to control the chemistry and allow us to understand what plasma process chemistry is actually responsible for what changes. So this actually was developed within the plasma grain project, and we've ended up with a means that we can functionalize liquids such as water to be a particular, to retain a particular chemistry for a particular amount of time. So we can actually introduce a function to a liquid through the cold plasma exposure. And this is one way of certainly knowing and understanding how it can be a biologically safe technology to apply for human interventions, but also for food processing interventions. So just to finish up, some of the other work that we're doing, we're really kind of pushing the boundaries and looking at new frontiers in biodecontamination across the agricultural systems, across food systems, and also across biomedical systems. So we have a range of ongoing projects. We're very happy to discuss collaboration. One of those is called EnvironSafe, which is looking at those allergens and also how we can modify and functionalize different food ingredients towards sustainable interventions to increase food safety. Plasma grain, as I've just gone through, is really looking at all grains and seeds, and we're moving on to look at the product development from a bakery perspective, but also from a fermentation perspective with the germination work, but also then on the agricultural application with using it as a seed pre-treatment, but investigating on a very holistic scale what happens with all of the different systems there. So we're looking at the soil, the water, the seed, the plant, the microbiome, the soil microbiome, etc. So that will hopefully lead then into some very interesting results in the near future. We're also looking at Campylobacter decontamination in the poultry industry, and this is in conjunction with other technologies and is also ongoing at the moment in UCT. So I'd like to say thank you very much for the opportunity again to present today. My contact details are there, and thanks very much for listening.